Guide RNA, gRNA, is a crucial component of the CRISPR-Cas system, acting as the GPS that directs the CA's enzyme to specific DNA sequences for editing. Here's a breakdown of its composition, structure, types, and functions. Composition and structure. A gRNA typically consists of two main parts, which can exist as separate molecules or be fused into a single molecule. CRISPR RNA, cRNA. This is a short, approximately 17-20 nucleotide sequence that is complementary to the target DNA sequence. This complementarity is what allows the gRNA to precisely find its desired location in the genome through Watson-Crick base pairing. The cRNA also contains a seed region, typically 8-10 bases at its 3 end, which is critical for initial binding to the DNA target. Transactivating CRISPR RNA, tracrRNA. This is a longer invariable sequence, around 65-85 nucleotides, that serves as a scaffold. Its primary role is to bind to the CA's nucleus, for example Cas9, forming a functional ribonucleoprotein, RNP complex. The tracrRNA often has specific stem loop structures that are important for CA's enzyme recognition and proper complex formation.
Types of gRNA. While originally the gRNA and tracrRNA were separate entities in bacterial CRISPR systems, for genome editing applications, they are often engineered into a single molecule, single guide RNA, or cRNA. This is the most commonly used format in CRISPR-Cas9 systems. It's a synthetic RNA molecule where the gRNA and tracrRNA components are fused together by a short linker loop. This simplification makes it easier to design and deliver the gRNA, leading to increased efficiency and ease of use. gRNAs are typically around 100 nucleotides long. Two-part gRNA plus tracrRNA. This tracrRNA. This tracrRNA. This is the natural form found in many bacterial CRISPR-Cas systems. While still used in some research, the gRNA has largely become the preferred format due to its convenience. Chemical modifications. gRNAs can also be chemically modified to improve their stability, reduce off-target effects, and enhance editing efficiency, especially in mammalian cells and for in vivo applications. These modifications can be added to the backbone, for example, 2-O-methylation, phosphorothioate bonds.
Functions of gRNA. The primary function of gRNA in the CRISPR-Cas system is to guide the CA's nucleus to a specific DNA target sequence in the genome. Here's how it achieves this. Target recognition and specificity. The cRNA portion of the gRNA with its user-defined sequence acts as the search probe. It forms complementary base pairs with the target DNA sequence, ensuring highly specific binding. CA's enzyme recruitment and activation. The tracrRNA, or the scaffold portion of the sgRNA, provides the binding site for the CA's enzyme. Once the gRNA-Cas complex locates and binds to the target DNA, which also requires the presence of a protospacer adjacent motif or PAM sequence immediately downstream of the target, the gRNA induces a conformational change in the CA's enzyme, activating its nucleus activity. DNA cleavage for gene editing. In the case of Cas9, the activated CA's enzyme then introduces a double-strand break, DSB, in the DNA, typically 3-4 nucleotides upstream of the PAM sequence. This DSB is the basis for various genome editing applications. Directed function, beyond cleavage. Beyond simple gene knockout via DSBs and non-homologous end joining, NHEJ, gRNAs can be used with modified CA's enzymes, for example nuclease-dead Cas9 or dCas9, to achieve other functions. CRISPR interference, CRISPR. dCas9 gRNA complexes can block transcription when guided to promoter regions, effectively repressing gene expression. CRISPR activation, CRISPR. dCas9 fused with transcriptional activators can promote gene expression when guided to specific regulatory regions. Base editing. gRNAs guide Cas fusion proteins, for example Cas9 fused to a deaminase, to specific bases, allowing for single nucleotide changes without creating a double-strand break. Prime editing. This advanced technique uses a gRNA fused to a reverse transcriptase to directly write new genetic information into a target site. Live cell imaging. DCS9 fused to fluorescent proteins can be guided by gRNAs to specific genomic loci, allowing for visualization of DNA in